Hey guys, welcome back. We are going to look at the end of aerobic respiration, really, for you and I, our big payoff, our big energy payoff. And so we've been through glycolysis, important but not a lot of ATP made. We've been through the Krebs cycle, important but not a lot of ATP made. And now we're really going to get to the point where we make a lot of ATP, and that is our phosphorylation. So a lot of ATP are formed during our final stage of aerobic respiration, and we call that stage electron transfer phosphorylation. You might have heard it called an electron transfer chain as well, which is fine. When you hear the word phosphorylation, right away you should be thinking ATP, because what do we phosphate and dephosphorylate? ATP, ADP. And that's what we're trying to do here. We're trying to take a lot of ADP and turn it into ATP, adenosine triphosphate, because that's a lot of energy. And that's a lot of energy that can drive cellular reactions. So again, we are still in the mitochondria, but we're going to change our location just a little bit from last time. And we're going to end up attaching a lot of phosphates to a lot of ADP to make a lot of adenosine triphosphate, ATP, make a lot of energy that way. So here's what this looks like. We've been spending a lot of time making NADH and making FADH2, these coenzymes that carry a lot of hydrogen. And now what we're going to do is we are going to donate the electrons that these guys carry, and we're going to donate the hydrogens these guys carry to our electron transfer chains. So active transport, this requires energy, forms this hydrogen concentration in our outer mitochondrial compartment. And then as the hydrogen flows back through, it's going to flow through ATP synthase, and ATP synthase is a very important protein that attaches a phosphate to ADP and actually makes ATP. And it's like a little motor or a little socket that rotates, making ATP. And the power to turn that socket is hydrogen ions flowing through. And so finally, oxygen will accept these electrons, and they'll combine with hydrogen, and it'll form water as our other product, because we formed CO2 earlier in the Krebs cycle, and we haven't formed water yet, which is the other half of that big formula for aerobic respiration. Now, this is pretty wordy, so let me show you a diagram that I think will make it easier. This one I like. NADH is a little yellow brick, and there's a bunch of them. FADH2 is a little brick, and there's a few of them, right? So we're getting this kind of 8 and 2 idea down a little bit. Now, these guys release lots of hydrogens and lots of electrons. Now, take a look at where we're located. Matrix, mitochondrial matrix, that is inside the mitochondria. The mitochondria has an inner membrane, an outer membrane, and an intermembrane space. And then out here is the cytoplasm and the rest of the cell. So know where you are. So NADH, FADH2, they're in the mitochondria, in the middle. And as they release these hydrogen electrons, we get a lot of hydrogens out here. But we have to force them through. Now, active transport requires energy. Most of the time, that energy is ATP, but not this time. This time, that energy comes from electron flow. So electrons flow through an electron transport chain, which is a whole bunch of transfer proteins that push these electrons through, and it powers the hydrogen gradient to get these hydrogens pulled through by these proteins. And so the hydrogen ions have to be pumped to a place they don't want to be. They don't want to be in the intermembrane space because there's a lot of hydrogens in here. And there's not so many hydrogens out here. So we have to pump them through with energy we get from our electrons. Now, once we have all these hydrogens in the intermembrane space, we have created a concentration gradient, and a steep one. And when there is a concentration gradient, simple diffusion tells us that ions will want to move from an area of high concentration to an area of low concentration. And as they bump along here, they can't get through this membrane. And they can bump along here, and they can't get through this membrane. The only way those hydrogen ions can get out of there is to flow through ATP synthase. And as they flow through, this little socket-like head rotates and rotates and rotates, and each time it turns, it grabs adenosine diphosphate, it grabs inorganic phosphate, combines the two to make ATP. And so we went from a place where we were just barely scraping by making ATP to the mother lode of ATP production, which is this electron transport chain. And so then at the very end, we have a little bit of oxygen left over. He grabs a couple hydrogens and becomes water. But from an energy point of view, he's not doing a whole lot. He's important, but he's not doing a whole lot for energy. It's really that hydrogen concentration gradient that is going to make that ATP. So all this time we've spent, transfer electrons, transfer hydrogens, transfer electrons, transfer hydrogens. We've got a lot of that. Make NADH, make FADH2 by transferring electrons, transferring hydrogens. And I keep telling you, these are energy nickels and energy dimes, and now you see why they're so important. Is we put all those hydrogens in here, and we make a ton of ATP. And so that's where this fits. Glycolysis flows to the Krebs cycle, flows to electron transfer phosphorylation, or the electron transport chain. And so the glycolysis products are the Krebs cycle reactants. Krebs cycle products are the electron transfer phosphorylation reactants. And it all flows together kind of beautifully to make one long chain of energy production that allow you and I to speak and move and survive. And so I'm a big fan of that. I like surviving. That's nice. So summary. This number, there's a little bit of argument about this number. Some say 36, some say 38, could be as low as 29, but it's a lot. You can break down one glucose molecule and get around 36 ATP, about as good as it's going to get. Glycolysis gets you about 2. Acetyl-CoA and Krebs gets you 2. Electron transfer gets you about 32. Okay? These two are preset. 32 can be a little variable, but a safe number is 36 ATP, which is a lot of energy. You think about, you take a bite of bread with all those starch molecules, and you break those down into how many, millions of glucose molecules make up that starch, and each glucose gets you 36 ATP. We're talking about a lot of energy of ATP coming out of this glucose. And so here's what it looks like. The whole thing all together. Glycolysis, we need glucose, we need ATP, our location is the cytoplasm. We have inputs, we have location, we have outputs. Outputs from glycolysis, NADH, it's going in the mitochondria. Pyruvate, it's going in the mitochondria. ATP, they stay out here, that's our outputs. Moving to the Krebs cycle, what's our input? When we turn that pyruvate into acetyl-CoA, acetyl-CoA is our input. The Krebs cycle makes its turn. What's our outputs? NADH, FADH2, a little bit of carbon dioxide waste, and the cycle continues with oxaloacetate. And then finally, electron transfer phosphorylation. We take all those hydrogens. Here's the inner membrane, right? We can see them spreading it out to show us as membranes. Hydrogens get in there. Electrons transfer through. We make ATP, and our water leaves the mitochondria. We end up making a bunch of ATP in electron transfer phosphorylation. And so there's your locations. Cytoplasm, inside the mitochondrial matrix, intermembrane space. Locations, inputs, outputs. There's a lot going on, but all of it working together is what fuels you and I. So we're going to stop right here for aerobic respiration. I want to show you a video that I think does a really nice job of carrying this flow of energy from our blood and our food all the way through. And so for you visual people, this could help a lot.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food. and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to...
Okay, so I want to pause it here for just a second. This animation is really good, but it shows them just they just sort of float in by magic, and bond with these carbons or take these electrons. I want you to bear in mind that each of these kind of opaque arrows is an enzyme. Represents each of those stages as an enzyme. So we have a different enzyme working here and here and here and here. They don't just float in. It's an enzyme that grabs two chemicals and puts them together, changes them and releases them. So it's very, very specific. They don't just float in making ATP by happy misfortune or happy fortune, just good luck. They're specific about these enzymes grabbing two chemicals and making this happen. So I don't want you to get too confused by the arrows and the weird floating glowing dots to miss the point that there's somebody in these arrows.
Understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain.
Okay, so there's a lot going on in this little diagram, but a couple things I want you to get out of this. These are all different proteins. They all have their different jobs. They're all transferring electrons, and they're all doing them kind of in sync with one another. And then what we get with this final protein is he gets a lot of electrons built up. That's a lot of free energy to be holding. And if we just continued to fill him with electrons, he would be very unstable, and it would really change his composition, and then we could be in bad shape. So having that free oxygen come in, take those electrons to form water, allows us to continue pumping electrons into the beginning of this chain and allowing them, all these different proteins, to work together to move those hydrogens through.
The bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
Okay, so I hope you guys found that video helpful. I'm going to post a link to another YouTube video that covers these same topics to give you just a different perspective on it, and maybe somebody who explains it a little more clearly than I do. So you'll certainly have resources available, but I know it's a lot to take in. So we'll talk about it more in class, and I hope this was useful. So thanks for your time.